Hello, welcome to week 3. Today I want to talk about source terms for SARS-CoV-2, the evidence for them, decay terms, persistence, and then we'll end up by posing a back of the envelope problem for exposure estimation. And I will pose the problem at the end of the first segment today, and at the second segment, work through the solution. So we've talked about this before. There are modes of transmission that may occur. There's direct contact when two people actually touch each other. And in this case, it's hand to hand. So transfer of virus from one person, the infected, to a susceptible. There's indirect contact by intervention of an inanimate object, a fomite. And then there's transmission in one of two ways: by particles emitted from the infected person to a susceptible person. They can be large particles that impinge directly on the recipient. The medical community defines these as 5 microns or greater and asserts, although we'll come back to this again and again, that it happens only at close range, less than 2 meters. Then the second mode of transmission is by finer particles that persist in the air for a period of time and are inhaled by a susceptible person subsequently. And that small aerosol transmission can happen at longer ranges beyond 2 meters. So I want to lay before you 2 documents that pose the controversy. WHO, toward the end of March, came out with a document on the left that asserted that SARS-CoV-2 could only occur person to person when the 2 individuals are close, in this case 1 meter, via coughing and sneezing and large particles directly impinging on the recipient. A couple weeks ago, a select expert committee of the National Academies of Science, Engineering, and Medicine were asked by the White House specifically to comment on the issue of what sort of particles in the air can transmit SARS-CoV-2. And this committee, in the second sentence, concluded that current available research supports the possibility that this could be spread via bioaerosols generated directly by patients' exhalation. And it goes on to talk about the difference between PCR and viable virus, as we've discussed previously. So this document supports the idea that small airborne particles can be inhaled by a susceptible individual. This earlier document by WHO refutes. And more, I think, and I would confess I'm on the side of the letter on the right, but more and more evidence and more argument seems to support that there is at least some transmission via small aerosols, which has an implication for us in terms of how we might control. The reason why this is important is that if you go into the early literature for the 6 foot or 2 meter separation, it really is based on the idea that in a stagnant air environment, large particles would settle out very rapidly. On the other hand, if small particle aerosol transmission was an issue, we would need larger separation distances. Personal protective equipment, such as masks, could be beneficial, both to reduce the source and also to benefit potential receptors. And ventilation and active air cleaning would help. The left-hand photo is a snapshot from a recent paper by a faculty member at MIT who did some very, very interesting high-speed photography of a cloud of particles released by an individual. Not an individual with SARS-CoV-2, but presumably a healthy individual, provoked a sneeze, and particles in stagnant air could be seen well beyond 5 meters, 15 or 18 feet from the source. Particles are released at high velocity, sort of as a jet, and even if they're large, they take time to settle out. So the issue of a 6 foot separation is really, at this point, dogma rather than science. There's also some evidence with SARS-CoV-2 from hospital sampling. Now, this particular paper is a preprint, and you should always take a critical eye at preprints in terms of who the authors are, and particular any comments on the preprint servers that are offered for review, because these have not yet been peer reviewed by journals. But these authors at the University of Nebraska Medical Center, which was treating a number of CoV-19 patients, did environmental sampling using PCR for SARS-CoV-2 in various locations. And particularly interesting to the controversy I posed on the last slide is that samples were seen at fairly high concentrations in the exhaust grate, the air grate from the room, indicating that at least RNA was in the air and was being withdrawn and impinged upon the grating. In addition, the presence in the vicinity of the toilet suggests the possibility of the virus in excreta. And then finally, air samples at low levels were seen as well. And so there is some evidence from hospital sampling that one can get at least longer range particles released from infected patients. Since this is RNA via PCR, we don't know their infectiousness. And now, even by ordinary breathing, and the next few slides are from Lindsay Marr at Virginia Tech, ordinary breathing generates aerosols in the small particle range. You see the peak here is just slightly less than 1 micron. Speaking, somewhat greater generation of particles, but again, 1 micron roughly as a mode. Coughing, perhaps somewhat smaller. But remember, if we say 5 microns is the so-called droplet value, in all of these curves, we have very, very few relatively particles being generated in that large fraction. Now, this was an intriguing study done on infected volunteers who were infected with influenza. And they were asked to recite the alphabet at 5 minutes, 15 minutes, and 25 minutes. And this is the total number of RNA copies observed within their breath, recovered in their breath, for 30 minutes. So essentially, their exhalations were contained in a device, and that total exhalation was sampled. For coarse aerosols, almost regardless of whether there's no cough or less than 1 cough per minute or greater than 1 cough per minute, about 3,000 RNA copies or 5,000 RNA copies were generated over 30 minutes. For fine aerosols, depending upon the cough status, there was a greater frequency of generation of fine aerosol RNA copies as coughing frequency increased. So no cough, roughly the same in coarse and fine particles, but as coughing increases, a greater fraction within the fine aerosol fraction of RNA copies. This is more work by the same group as in the previous study who looked at the question of masks. And what's interesting here is you see, and of course, this is in accordance with WHO and CDC recommendations. For coarse particles, we get very good reduction in terms of RNA in the exhaled breath if the subjects were wearing a mask. For fine particles, maybe only a 50% reduction, because regular masks, even surgical masks, are not designed as barriers against the fine particles. Of course, for N95 masks, which are designed to remove the fine particle fraction, we would expect a greater degree of removal with the mask. Now, how about for coronavirus? So this is a paper that was done, but with seasonal coronavirus. And there's a lot of information on the paper, 3 different types of virus: coronavirus, influenza, and rhinovirus, which is the most frequent virus for common colds. And the bottom line is you do get good reduction of the large particles with masks, but you still get some release, even with a mask, of the coarse particles, and of course greater with the fine particles. Now, one of the reasons I put this figure up is that it tends to show that there's similarity in behavior amongst these three viruses, seasonal coronavirus, influenza, and rhinovirus, which leads us to suggest that maybe CoV2 would be similar in behavior as well. Now, after the particles are released in the air, they can decay. And the decay rate is a function of temperature, but it's also importantly a function of relative humidity. And so this is work by Professor Marr and one of her students. What's plotted here is the decay rate. So as the decay rate increases, that arrow is incorrect. Decay rate is actually increasing toward the bottom. As decay rate increases, the virus lasts less time in the air. And so humid air is good at increasing the rate of inactivation. Now the virus is released. It survives in the air, whether it's a large droplet or a fine droplet. How is an individual exposed? Well, the respiratory tract contains different regions. Broadly speaking, there's the upper respiratory tract and the lower respiratory tract. You see coronavirus up here at the upper respiratory tract. That's believed to be the primary site of infection, which is why nasal and nasopharyngeal swabs have been used for sampling the virus in suspect cases. Other viruses infect the lower respiratory tract. On the right-hand side represents the fraction of particles of different sizes that are inhaled as a function of the amount retained in the lungs. And the region that we're talking about for the upper respiratory tract, the head and nose, to some degree the tracheal bronchial. And you see almost for every single compartment, there's a particle size at about a half micron, give or take, that represents the minimum retention, the minimum deposition. As we go to greater particle sizes, we get a greater degree of retention. Smaller particle sizes, a greater degree of retention. Sounds a bit peculiar, but what's happening here is that at the larger particle size, we get almost gravitational sedimentation onto the walls of the various components of the respiratory tract. On the smaller sizes, what's happening is Brownian motion starts to occur. And Brownian motion is when we have a small particle, but by the action of molecular forces and collisions with air molecules, in this case, in the airway, the effective diameter becomes larger. So in essence, these small particles present a greater effective area for contact than their physical size alone would dictate. Now, the material in which the aerosol is emitted into the air may have an influence on the decay rate as well. This is more work from Lindsay Marr's lab. The previous line that I showed you was when the virus was suspended only in salt. But you see if protein is present as well, which we would expect in an actual aerosol from an infected individual, from saliva, from mucus, and so forth. At high humidity, the decay rate decreases again. So large humidity is not beneficial, and the sweet spot, again for influenza, looks like about 80% relative humidity to maximize the decay rate. Now, how about SARS-CoV-2? How does that survive? And data are only starting to come out on the current virus. And this paper by Van Doormalen et al. In New England Journal of Medicine is hot off the press. But it basically shows that the half-life for aerosols of SARS and SARS-CoV-2 is roughly similar, with a half-life of about 1 hour. Half-life on surfaces depends very much on the surface. Longer half-life on plastic than on stainless steel. More of a difference between CoV1 and CoV2 on cardboard. More of a difference between CoV1 and CoV2. And copper die off very quickly. And this number has certainly been the source of a lot of press. Let's say a 3-hour half-life on cardboard. So question is, how much of a risk does cardboard and touching it have to people, for example, that get package delivery and so forth? Now, I want to refresh your memory between the relationship of half-life and a rate constant. Essentially, by invoking the concept of a half-life, we're saying that die-off follows an exponential decay. And it certainly may for a while, but whether it does entirely is not clear. We characterize the exponential decay rate by a rate constant K, and there's a relationship between the rate constant and the half-life. Now, getting back to the issue of surfaces. So if we look at a scenario where a surface is contaminated by some mechanism, maybe by a handler with unclean hands, maybe by a cough or a sneeze by an infected individual, and that surface is then contacted by a second person. So we have transference from the fomite to the hand. And we need to describe how well that occurs. We then have transference from the hand of the person who had contacted, or from the hand of a person who may be infected, contacting the surface. And then finally, we have contact between the hand of a person who's just touched the surface and their own mouth or nose or eye. How much of what they've touched now are they exposed to by contact with a body part? So in the next 2 slides, I've extracted some data for you and some references. And people have measured this for a variety of viruses. Unfortunately, not yet for SARS, but rotavirus, You see Ansari et al. Measured 1.8 to 17% surface to hands, 2.8 to 6.6 hands to surface. Mabithi et al. Did measurements on Hep A, and with Hep A, they applied the virus to the surface in a wet suspension, and transference declined as drawing occurred in both directions, surface to hand and hand to surface. Rusin et al., and this is one of the few data sets that measure transference from fingers to mouth, because recognize there is significant biosafety and human subject issues if you were to do the experiment with a known pathogen. And they examine for phage, and remember, phage are viruses that infect bacteria. About 34% transferred from fingers to mouth. Julian et al. Looked at 4 different phage and did a very large study with more than 600 volunteers looking at transference between fingertips and glass to fingerprints. And the take-home message from this entire very large study is, for all the bacteriophage examined, whether it's transference from glass to finger or finger to glass, roughly 30 to 35 or 40% transference. So I want to take a break now after I pose the problem. And the next segment will be solving this problem. But let me pose the problem and take a few minutes to think about this, or some time to think about it, sketch out a solution, and then listen to segment 2 of this week's talk. So let's say we have a scenario in which an infectious, contagious individual let's call them person A, is in an enclosed room for 30 minutes. There's no ventilation, and the person is not coughing. Now, I'll come back to this issue of ventilation at the end. Next week, we'll talk about what happens when we have ventilation involved. But this is a simple problem, assuming that the air is totally enclosed and that any viruses that are mixed get completely mixed with the volume of air. The room is 5,000 cubic feet with an area of about 700 square feet, maintained at 50% relative humidity. All coarse aerosol deposits on a table that's 8 foot by 4 foot in area. An hour later, after person B has left from her 30 minutes, an hour after that 30 minutes, person B comes in, stays in the room for 15 minutes. Person B does not use a mask. Person B touches the table once and then touches fingers to nose and is obviously breathing the entire time. So work out what the total exposure is to SARS-CoV-2 based on information I presented previously this week from the fomite and from inhalation, and then we'll go ahead and talk through the solution.